successful in controlling FMD, and definitely in Europe, where there's currently no FMD, but there was, and they were actively vaccinating up into the 80s. They're no longer vaccinating. And then even also in South America, that still does have some FMD in some countries, but they've been quite successful in controlling it. Problems with the vaccines themselves is that the duration of immunity is very short. So in endemic regions, they need to revaccinate at 4 to 6 month intervals, which is quite a lot if you have a lot of animals. And then there's this additional thing that's inherent to the virus, is that there's so much variability. There's serotypes, but there's also variability within the serotype. So in order for your vaccine program to be efficient, you need to match your vaccine with what you believe will be coming through, basically. Currently, there's this official difference in status of countries that are vaccinating to control FMD or that are free without vaccination. And this has a massive impact on the ability to trade in animal products. So that many countries have quite a far way to go still to control the disease.
You mentioned about the duration of immunity. What's known about, what makes it so short?
I don't have a good answer for that, but that's definitely something. There is a lot of research done into vaccine and specifically developing different platforms of FMD vaccines. It's just not quite on the market yet. I don't really have a good, the sort of holy grail there is if you have natural infection, we believe that the immunity is very long for that specific virus. That's just not the same when you use inactivated vaccines. I don't really have a good answer to why. But right. It's definitely an issue.
Okay. No. Yeah, I guess I'm thinking of, polio, right? Another coronavirus, obviously, yeah. But you just mentioned that in natural infection, the immunity is quite long. I'm thinking that another factor that might, and this is, I'm sort of tapping into some of the questions that come through about shedding in feces, which then, I understand it is pretty high. Is that right? Or no?
So in contrast to other picornaviruses, FMDV is extremely acid sensitive. So it does get inactivated through the gastrointestinal tract. So at least as far as we know, fecal shedding is not important for disease transmission as it is, for example, with polio, which has a totally different resistance.
Right. And are there, I guess, there's environmental stability? I guess the virus is reasonably stable.
Yeah, it really depends on both the pH and the ionic composition of whatever material you have. So in certain circumstances, it can be durable in the environment. It really depends on the conditions, but there's a lot of people that are actually working in just environmental sampling to just as a surveillance, that you can go to animal markets in endemic regions and basically sample the environment to get an idea of what viruses might be passing through.
Right.
So there's some applicability to that.
Okay. I guess we'll spread around the wealth here. So I guess, Amy, I guess I got a question for you. I've got a couple of them. I guess do you know the effects of the in utero infection of Rift long-term on the rat pups that survive? I guess you want to talk about that a little bit? Just are there any long-term effects?
That's a really good question. It's one that we haven't yet had a chance to explore, but we would definitely like to. So the pups that survive, we know at the end of our study, at about 18 days post-infection, that the pups have viral RNA in them, but we don't know what developmental effects that will have if we let the pups develop. And we definitely want to do that. And so that's those studies will be ongoing.
Mm-hmm. I have a question that for you, Amy, but I guess Dennis, this might be appropriate for you as well, which is, salivary gland, the extract proteins in the salivary glands of vectors, can potentiate infection. I was just curious if you have plans to look at that, maybe at low dose infections in your animal models.
It's not something that we have done previously, but it is something we could do in the future. Dennis, maybe you have more information on that.
Yeah, it's critical for tick-borne diseases. One of the transmission pathways is what we call co-feeding transmission or non-systemic transmission. So an infected tick can infect a native or non-infected tick feeding in close proximity or on the same animal, even if there's a systemic immune response, if there are antibodies or T cell response or so on. And we know that this co-feeding transmission is enhanced due to the saliva of the tick. There are also some other studies with Powassan virus and so on that show that the saliva really enhances the initial viral replication in the replication side and then the systemic spread. So very crucial. We've done a little bit of work and mostly with explants and cell culture work, a huge component that's often not included in animal work, right? We always do needle inoculations. There's hardly any studies that involve tick saliva in those studies.
I would imagine if you did, it could be masked, by having, if the inoculum is too much, whatever potential benefit with salivary gland extract, it might be masked if your inoculum is too high.
Yeah, exactly. So what we've shown with studies that we did on Langerhans cells and dermal dendritic cells is that the saliva exactly does the same thing. So it will not necessarily enhance the virus replication, but it will mask the immune response. So the virus can get into the cells, can replicate, but the cells will look like they are not infected. They are in a non-activated state. So the tick saliva will suppress the immune response.
Okay, interesting. Let me see here. I guess a question for for Dennis. I got one more for you. I guess, do you see foresee the introduction of Hylomatix in the U.S.?
That's a risky question. What do you want me? It's a lose question for me, right? Like if it's not happening, then I was wrong, and if it happens, I jinxed it. And
Right.
I think we see a lot of non-native tick species being introduced into new areas, and you don't necessarily see that they establish in certain areas. I think so. It takes a lot of different things. That was also one of the questions. So you have to import the tick. The tick has to be imported into the country, but the tick also has to find the right animal hosts. So it's not only about temperature and humidity, but it's also finding access to the right hosts. And so I don't think it's as easy as maybe like what we see with the mosquito-borne viruses.
Mm-hmm. Well said. I guess I had a question for you, Jessica. I guess, can you talk a little bit more about the neurological sequelae for NEPA and how these models can be used, the models that you presented can be used to investigate them?
Sure. One of the things that I didn't have time to mention in the talk is in our hamster model, we've seen something that's a little different from some of the other labs in the sense that we have animals that have neurological signs that they actually recover. So we've had animals that have had pretty significant head tilts, almost 90 degrees, but we monitor them. And as long as they're maintaining weight, they have access to food and water, we'll follow them. And some of these animals have recovered in the sense that by the end of the study, their head tilt is getting better, if not back to normal. Whereas when I was talking to some of my colleagues that have worked with the model previously, normally when they see onset of neurological signs, it's just a downhill slope from then. So what's interesting is for NEPA in particular, the neurological sequelae can be quite long-term. So they have a late onset phenotype, which can also be in humans that have had mild to asymptomatic infection. This was studied in one of the outbreaks. And you can also have relapsing. So cases that had recovered from neurological signs earlier that have later signs. And a subset of these cases are actually fatal when they reoccur or when you have late onset. The other thing is when they were following survivors, there's changes, kind of similar to what we've seen with COVID, is they have behavioral changes, concentration issues. There's been discussions of ataxia, so motor skill issues. So it's quite significant in this case. And the model, as I said before, if we have the opportunity to really follow some of these animals, whether they had clinical signs or not, and kind of striate in groups, and see if we can recapitulate some of these phenotypes that are seen in humans and do some advanced imaging and other fancy techniques to try and understand if there's actual virus still there, or if this is just like a secondary effect. I think that would be incredibly interesting and valuable.
This is a question, I guess, about ticks in general. So I guess whoever wants to jump in, feel free. There's an interesting one that came through, which was about what would be the potential repercussions of eliminating ticks. Say, we'll start with hyaloma. You know, I guess, do ticks do any good? I guess is the other way of looking at it.
Yeah, that's, I don't know if anybody else. I mean,
Yeah.
I was going to say, you have a love of ticks with that big background. I feel like you would be sad if they went away.
Right. Well, I get that question a lot, right? The question is always like, do you influence the biodiversity if you remove ticks from a certain area? You know, I disagree with that. It's not a key species. When it comes to biodiversity, we have these food chains and so on. And sure, ticks serve as food for certain birds or something like that, but they don't really play a key species in these food networks that we see that are, you know, with the mammalian species is much more complex. You also see a lot of dynamics. You see that like ticks appear in new areas, then they disappear. You know, if you look at what's going on with the mosquitoes, that's not a big discussion with mosquitoes, right? Like nobody cares about the biodiversity of mosquitoes being eliminated in certain areas. So I don't think that we would see an influence on the biodiversity and an influence on the ecological networks if we remove ticks.
I guess, Carolina, a question came through, which I'll pass on, which is can we make a more general assumption that ruminants fail to clear virus and non-ruminants do clear it?
Based on what we know, that seems to at least partially be the case. There has been some experimental infections done in other types of ruminants, like wildlife, different types, species of deer that we have in North America. And as far as I know, those did not maintain persistent infection to the same degree that cattle do. There's also a difference between sheep and goats as small ruminants. They do become persistently infected, but usually for a shorter time period than cattle. So I'm not sure we can say like definitively that there's like a definitive difference, but as far as we know, we've only seen persistence in ruminant species, and then it might be a little bit far to say that all ruminants become persistently infected.
I guess you could use that in general terms, that difference to try and figure out the mechanisms of
yes.
clearance or
yes.
not, I guess.
Definitely.
Yeah. Okay. Let me see here. I guess, Amy, I got a question for you, which is the trophoblasts in their rat model. Did they also become infected?
They do, yeah. So we've done in situ hybridization as well as fluorescence microscopy with an anti-rift antibody, and we can show that the trophoblasts in rats, which are different named cells than what we have in humans. So they're called giant cells, and they're called spongotrophoblasts, and they do get infected. They do get infected very highly, actually. And so those are the probably most interesting cell types that we're looking at, trying to figure out, you know, why are these cells so susceptible to rift infection?
I have there's another rift question for you, and that is, I guess the wild type seems to significantly affect fetal development in many cases, and so based on that, I mean, and should we, as a field, start moving away from live vaccines and more towards subunit or vector vaccines? Well,
I think for humans, probably yes, if we're going to go down that route of a potential human vaccine. But for livestock, the live attenuated vaccines seem to be a lot more practical, but it would be worth looking into whether either subunit type or vectored vaccine could be as effective in livestock with one shot as the standard live attenuated strains are, because that's the thing is that for livestock vaccination program, you really need a one-shot deal. so as not to have to come back and revaccinate the livestock herds. So I think it is definitely worth looking into.
I'm trying to remember for the DLNSS RIF vaccine, it did not go into, it didn't go across the placenta, I don't think.
Right, yeah. So what Dr. Towner is referring to is one vaccine strain developed at CDC is the del virus, where it's deleted for NSS and deleted for NSM. And that when they did a really nice experimental study in pregnant sheep, they did not see transmission across the placenta. So that is very promising. Now, I don't know currently where that vaccine is in field trials, but that is the one live attenuated RIF vaccine that has not been shown demonstrated vertical transmission.
Right. I guess while we're on the subject of RIFT, I guess, Amy, there was a question in the chat box. I don't know if you answered or not, but it was about ongoing RIFT surveillance programs in women in Africa. Any thoughts on that? Or
yeah, so part of the reason why I like to get out and talk about our data is so that people that are working in Africa, in epidemiology, in those types of areas, which is not my area expertise, but I really think there needs to be a renewed focus on pregnant women so that when there is activity of RIFT in livestock and people, that data can be collected on pregnant women, because it's gone completely overlooked, right? In general, pregnant women get overlooked for a lot of things, right? Vaccines don't get tested in them, therapeutics don't get tested in them. And then in general, I think they've just been overlooked when data has been collected on RIFT outbreaks in the field. So I hope that people will focus on that now and try to understand, okay, what is the effect in these women? Have they gotten infected during pregnancy, and can we collect additional data on that? So that's one of the things that I hope will happen by talking more about this potential effect in pregnancy.
Let me see here. I guess a question for Jessica. Just trying to spread the wealth here. You talked about the sequelae for Lassa and NIPA, the long-term ones. You might chat a little bit about that for CCHF?
Sure. So I actually messaged Dr. Bente last night to ask him, we were chatting about this because I'm not aware of any reports of well-described sequelae for CCHF. And to me, given the fact that we know how widespread it is in the tissues, again, kind of based on recent experience of, the pandemic virus, that we know when it's hitting other tissues, there are bound to be consequences of that. So there have not been studies into this. And I didn't know, Dennis, in your time working so Turkey has a large number of cases relatively to other countries with low case fatality rate when it comes to CCHF. So that would really be a really good area to potentially follow patients and see if there's any implications from infection. But then also some of these more severe cases, if it does really affect the liver and we've seen changes in clinical parameters, I can't imagine that there wouldn't be any implications of that type of disease. So I'll open it up to other panelists too, if you have thoughts.
Yeah, I can only confirm what Dr. Spengler was saying is, what we've seen in Turkey, where there are quite a number of survivors. There are long-term sequelae, but I think they're often very unspecific and probably not related to a CNS infection, as we've seen maybe some, tick-borne encephalitis infections. So it might be some collateral damage of the actual infection. So there are some sequelae, but they're not very well characterized.
Have a question for Dr. Spengler. Given the utility of looking at varying strains of some of these segmented viruses, have you looked into the strain segment reassortments in animal models that offer viremia, non-lethality, persistence, et cetera?
Well, we've done a little bit of work with that. We used the guinea pig model actually to look at Lassa, and I mentioned the Josiah and the New Jersey isolate that we had used in the eye studies. So actually, in some of those studies, we made reassortant or recombinant viruses where we had swapped the two segments, so the S and the L, to try and determine, which was contributing to the pathogenesis or the lethal phenotype. And what we found, similar to what was described for the ML, there's a vaccine that's based off of swapping Lassa and a pathogenic arenavirus, it was the S segment that was contributing to actually that lethal phenotype. So we have done some of that work.
Right. I guess this question probably for Carolina that came in through the box. I guess, could a tick, this is less, but anybody can chime in on this, is kind of a bioterrorism kind of a question, because could a tick infected with a foreign animal disease be used to intentionally introduce a disease into the US? And I'm assuming the answer is yes. That was a question for Carolina.
Well, I'm not really into the tick.
Okay.
And so I think that might be Dr. Bente might be a better person to answer that question.
Yeah, that question is the bane of my existence, because every tick species that live ticks that we import into the US, exotic tick species, has to be screened for foot-and-mouth disease, if it comes from a different area. If you look at the literature, there's never really been a described case of a tick carrying foot-and-mouth disease. And, I worked on foot-and-mouth disease in a previous lifetime, so it's not really my wheelhouse. But from what I understand is FMD is not really viremic in animals, so there's less of a chance that the tick will really acquire the virus through the blood and when it engorges. There's been a case recently described, I think in 2006, if I'm not wrong, where a tick from Kenya that fed on cattle was imported into the US and was tested positive for foot-and-mouth disease. So that kind of like shook the general perception that ticks, when they acquire blood meal, cannot carry a foot-and-mouth disease. I think it would be more of like I don't want to call it a mechanical transmission. I don't think there's a biological replication of foot-and-mouth disease in ticks, so they will not give it to the next life stage. But if you collect ticks from livestock in FMD areas and you import those ticks into the United States, there's definitely a chance to import foot-and-mouth disease.
Yeah, so just a comment on that, based on FMD, it definitely is viremic, but it's not really I think if tick-borne transmission was a factor in disease transmission, we would have seen that in regions where FMD is endemic. And I think, like Dr. Bente says, it might be that the tick can mechanically carry the virus, but I can't really, FMD is very specific with which cell types it replicates in. And I can't really see that how it would transmit it. I mean, it's hard to prove a negative, right? Based on what I know of Hylomatix virus, I don't really believe that would be a substantial pathway. But then again, there's a lot of foreign animal diseases, and it's hard to make a blanket statement and say that something can't happen.
I guess still on the subject of the ticks, there was a question that came in earlier. I was also wondering about is, what efforts are underway to make vector incompetent ticks? I mean, you can use like CRISPR-Cas, to make a incompetent vector. I mean, you see this with mosquitoes and mosquito-borne diseases where you're introducing sterile, insects or mosquitoes. Are there any experiments underway along those lines to make incompetent tick hosts?
Yeah, this is a very good question. It's always been a technical issue to genetically modify ticks. So when you do embryo injections, what you do with mosquitoes or something, the shell of a tick is so thin that it will just explode if you inject something into the egg. And this has been technically challenging. Another thing is that the genome of many tick species is not fully annotated. There's been lately a breakthrough. A group in Reno has been able to genetically modify ticks. That's actually one of the things we started collaborating with this group, and we hope that we can genetically modify ticks in order to make them incompetent in transmitting some of the viruses that we study.
A question here for Carolina, I guess what are can you maybe talk about the different pathogenesis phenotypes for the 7 different serotypes of FMDV?
Yeah, I think we briefly touched on that before, and there are 7 different serotypes. There's also a variety of different strains within the serotypes, and the different serotypes are differently variable. We do see difference in pathogenesis and virulence between strains. It's not necessarily governed by the serotype. It is one of these things that's hard to address experimentally, because on one side we want to standardize our experiments. We want to use the same virus and the same type of host. And if you want to establish like parameters of disease phases, you can either choose to get as strong data as possible for your selected strain, or you can choose to get a broader set of data for covering different strains, but then you won't have the same power in your investigation. So we try to usually end up somewhere in the middle, that we try some different viruses, but it's hard because there are so many types. It's hard to, we can't test all of them basically.
Right.
But there's differences both depending on the virus strain and then the host species and the combination thereof. So different strains will have different virulence in different species. But again, it's more at a strain level than at the serotype level.
Mm-hmm. Any differences in stability between this?
I've actually been looking into this recently. There's this conventional wisdom that one serotype, specifically O, is less stable, and that it's somewhat consistent to what we see. We've been looking at specifically viability of FMDV as a potential feed contaminant. I've had trouble finding references for this in the literature. I think it's something that specifically people that work with empty capsid vaccines, they know that serotype O is a little bit more tricky. But in general, all of the serotypes are way less stable than most picornaviruses, specifically pH. And that's just a part of the mechanisms of the cell entry, that the capsid is basically supposed to fall apart in the endosome. And that's just how the virus works.
Right. I'm curious, is there I presume there's a reverse or genetic system for FMD?
Yes.
There is, yeah. And then you could look at that specifically, yeah. Let me see here. What other questions we got? I guess staying on with the FMDV, does the persistent infection in the carrier state, I guess, what are the implications of that for disease control, I guess, overall, if you want to maybe comment on that?
Yeah, it's a tricky question. It depends on who you are and what your purpose is. If you're in an endemic country that struggle a lot with clinical FMD, then persistent infection is probably not what you should be most concerned about, that you need to control the clinical disease by vaccination. On the other hand, if you're a country like in Europe or the US, where you don't have foot and mouth disease and you have an incursion that you need to control, then you need to be able to prove freedom of disease, which means that you can't have any persistently infected, regardless if they're sick or not. You can't have the virus in any animals. You need to prove freedom of disease. And then again, it's this thing, it's hard to prove a negative. We have in limited experimental studies, when people have tried to study transmission from carriers to naive recipients, no one has been able to prove that it actually does transmit. However, we can pull infectious virus out of these animals and these throat swab samples, and we have done that experimentally. We've taken this throat scraping, we call them probang samples, from the upper respiratory tract of persistently infected animals and just mechanically transferred that to naive animals, not injecting it, but just basically putting it into their nose, and they do get sick. Out in the world, there's a lot of foot and mouth disease, and after each outbreak, there will be a lot of persistently, I mean, there are millions, I don't have a number, how many persistently infected carriers there are in the world. And it's a little bit hard to take a step and say, like, they don't matter. And what we've seen experimentally recently, this is work in progress, we haven't actually published it, is because the limited protection between the different serotypes, so you can, if you have a persistently infected animal, it can become infected with a different type of the virus. And if that happens, we actually see viral recombination happening in the upper respiratory tract too. So, and when you do that, when you have these sequential infections, the second infection will actually promote virus shedding, because then again, you have a persistent infection combined with an acute infection. and whether or not the animal becomes sick, they will start shedding virus, and you have the potential of these animals actually functioning as sources of new recombinant viruses, which is something we've seen experimentally. We haven't had the opportunity to really get into it in the field, but we know in our field sampling that we do find a lot of recombinant FNDVs. So it's possible that these animals do have a role to play there.
Are there any sort of predictable host changes? I'm just thinking, like, there's questions about like this comes up in the bat world, about recrudescence and pregnancy when you have zero hormonal changes that might cause a persistently infected animal to start to shed more.
Yeah, it's a good question. It's again something that's very hard for us to study in the lab because we can't really have pregnant animals. There has been hypotheses that it's the other way around, that it's the male animals that somehow gets provoked to shed virus, and those hypotheses are out there. I don't think there's any solid evidence either for or against it, but this is one of those things that's probably easier to study in the field than in the lab.
Right.
If you have the opportunity.
Yeah, and I imagine one of the issues in terms of overall control, at least my own experiences, oftentimes you have livestock that are right up next to wildlife, buffalo, for example, right? And you got these boundary areas where it's just hard to, you kind of have these continual sort of reintroductions of virus.
Yeah, that is a problem. I mean, there are studies of that specifically. We've done some field work too of sampling cattle and buffalo in Africa that share some areas, and it doesn't seem to be it seems like the viruses are mostly specific for each of the species, but there is some cross-species transmission going on, definitely.
Right. I guess, Amy, I got a sort of a general question for you. I guess for your overall presentation, I mean, what are the sort of the most pressing, questions that you want folks to take away from your comparative placental studies?
From my general talk, I would just like everybody to take away the idea that pregnancy and vertical transmission with regards to Rift Valley fever really should be on the radar of clinicians and epidemiologists and field workers, so that we can understand better, does this occur, how often does it occur, and what are the outcomes. I think with the comparative placental studies, I think what we're really hoping to understand is common mechanisms of vertical transmission that occur across the different species, whether that is some sort of particular cell type that's targeted, or attachment factor or receptor. These are the types of things I hope we can learn from the comparative studies.
Thank you. And I guess maybe a brief comment on why do you think placental studies in general have been an unrecognized target?
For Rift Valley fever or in general?
For Rift.
Yeah, that's a really good question. I think from an experimental standpoint, I think that the studies just weren't done, right? So nobody took a pregnant rat or mouse and infected them. There was one study that was done back in the early 80s that infected pregnant mice. And it described very briefly that it it did not describe fetal deaths, but did describe viral RNA in the embryos. But other than that, the literature was basically nothing when we sort of set out to do this. And at the time we generated this idea, the Zika outbreak was, kind of winding down. So that sort of emerging mosquito-borne disease that can affect pregnant women was on everybody's radar. And so it seems sort of like an obvious connection. All right, we know Rift Valley fever does this in livestock. Could it do it in people? Probably high likelihood. And then can we develop a rodent model? And I will say that the difference between the rodent models of Rift are that, we don't have to use an immunocompromised mouse. So a lot of the Zika studies, you have to use immunocompromised mice in some way to get disease. And even the pregnant Zika mouse studies, they usually infect the pregnant dams early in gestation to see the greatest effect. And we did both. We infected early and late, and we ended up following up with the late gestation because it seemed to be a little bit more relevant, right? And if we're seeing these effects late in gestation, then I'm sure they would be even worse early in gestation when the placenta is not even formed yet. So yeah, so did that answer your question?
Yeah. Yeah.
Can I ask Amy?
Yeah,
So
sure.
given the nature of disease in a lot of these rodents, you know, how it can be quite acute. Do you think that that was limiting also some of these studies in the sense, it kind of piggybacks on my own point of the day, but.
Yes, right, yeah, no, Jessica's point is well taken, right? So rift infection in rodents and especially mice is really acute. You know, the mice will start dying of 2.5 days post-infection and they're all dead by 4.5 days. So it's a very short time frame. And so, yeah, I think that has a lot to do with it. Like a lot of the work that's been done with rift in mouse models has been this acute hepatic disease model. That's why, you know, when we started looking at viral encephalitis a few years ago, you know, we had to use different strains of rodents in order to get that outcome. Kind of like what Jessica was saying, it's like we need to make sure that our animal models are reflecting in some way, you know, the course of disease in people, right? And we can have different models modeling different clinical outcomes, and they'll all teach us something important. Although no model is perfect, we can still learn stuff from them.
As I say, if any other panelists have questions, feel free to chime in. I guess I got one question, because for Dennis, just in general, and this encompasses a lot of different factors, but you know, for CCHF, I mean, in humans at least, you can have you know, those viremia become a lot of virus shedding, but you don't see the longer chains of human to human transmission like you do with filoviruses, for example. Any thoughts on where they're, you know, why that doesn't happen?
Yeah, it's a tricky question. I think that's, you know, and please correct me if I'm wrong when it comes to Ebola, but I think we do see high viremia levels. That's correct. And we do see transmission in a hospital setting. There's some kind of a procedure that's produces an aerosol like intubation or something like that. But I think generally speaking, the shedding from the mucous membranes is maybe not as intense as we see with some of the other hemorrhagic fever viruses, right? Like I assume that maybe, you know, with Ebola, you see diarrhea, you see, you know, a lot of high titers in the saliva and things like that. You don't see that too much with, you do see diarrhea with CCHF in some cases, but I don't think that that's really a mode of transmission or a crucial mode of transmission. It is in the bloodstream, and also keep in mind by the time the main symptoms set in, the virus is already being cleared out of the system. So I'm speculating here.
No,
I think it's mostly bloodborne and less mucosal transmitted. And Jessica might be able to.
I don't know. All I was going to say is that I think we, you know, until something changes, you kind of are in this mode that, like, even with Ebola, right? No one would have predicted what happened, you know, a few years ago, even though we did see larger clusters than what we've seen with some of the other pathogens. I think it's a matter of circumstances and many other things that are hard to predict. So whether or not there may be larger outbreaks with CCHF or whatever, I think that remains to be seen. And that's why we have to be as prepared as possible in the pipeline. I think we were fortunate with Ebola because that was arguably the most advanced we were with therapeutics at the time of that outbreak, but that's not the case with many of the pathogens that have been discussed here. So just to keep that in the back of our minds.
Yeah, I agree. I think Jessica's absolutely right. I think just the nature of Wunya viruses, there's just not a lot of human to human transmission. Andes is one of them, CCHF. There's now speculation that SFTS might also be transmitted person to person, but there's very little evidence. So I think there might be a chance, but it might be not as intense as it might be with some of the other hemorrhagic fever viruses.
There's a question that came through in the chat box, so I guess it might be for you, Dennis. It says USDA-APHIS PPQ uses a sterile insect technique to control Mexican fruit flies, fruit fly outbreaks. Could this be used on ticks?
Well, to be honest, I'm not 100% sure what technique. I assume they use irradiation. They irradiate the male fruit flies. If I'm not sure if that's true or not. There's been some work done in the 70s and 60s where male ticks were irradiated and then released to see if that has an influence, and that was not very successful. I don't know why, but there have been attempts decades ago, and it's just not successful, maybe because like a female can mate with multiple males. I don't know. I'm not sure. That's really not my wheelhouse.
Right. Yeah. But I guess with like TCHF, you have the virus basically transmitted through the different nymphal stages, right? And so that would be what you wouldn't have with your fruit flies, I guess. So, I think any other, I'm kind of out of questions here. Yeah, I guess maybe Dennis, you might maybe mention a little bit about some of the constraints working with ticks in maximum containment. I mean, what are the precautions you need to take? I mean, clearly they're small, it can mobile a little bit. So maybe you might chat a little bit about what it's like to work with ticks in BSL-4.
Sure. I mean, the main constraints are due to the select agent regulations that we have. So, if an animal is potentially exposed to a select agent, everything has to be tracked and traced. And the same applies for the ticks that we work with. So we have to maintain accurate counts when we infect ticks, and that might be easy for adult ticks, but it's very tricky if you have the immature stages that are very difficult to count. And plus, when you feed them on animals, there's always a certain number of ticks that will just desiccate and die, or get smushed by the feeding capsule. So what we're doing is we're using multiple layers of containment. So we restrain the questing of the tick by using feeding capsules. They are tailored to the tick species, to the animal species. So on a mouse, it might be like a small tube, like a Sarstedt tube or something that sits on the mouse. And rabbits, we use soft sock-like feeding capsules that go onto the ears or maybe onto the back. And those capsules are very secured. That's our first barrier that we work with. Then the second barrier is the cage. So we use a lot of sticky tape or Vaseline. Tanglefoot is a sticky type of Vaseline. So our cage is the second line of defense. So in case the feeding capsule breaks and the ticks are released into the cage, then we see the cage as a second layer of defense. Again, sticky tape, it's a sealed cage. It has white bedding in it, all of those different things. And then we have a specialized tick room that also has a lot of barriers, sticky mats, moats of sticky Vaseline, things like that. That's our third barrier. And then obviously we have the BSL-4 as the fourth barrier of everything. I don't know if you saw that in my presentation. When we work with the tick work has to be done in a glove box. So in addition to your positive pressure suit, you also have to reach into a glove box and unscrew so if you open primary containment, that has to be done in a glove box that also has sticky tape and all kinds of layers. So we really take a lot of effort, precaution that those ticks can't escape. And that makes the work very tedious. People always think it's a dexterity thing, but the dexterity in the suits is actually fairly good. And the tools that we can use to visualize, we have loops and so on that you see from the dentist, at the dentist office, these loops. So we do have, dexterity-wise, I think we're in a good place. It's just more like these barriers that we have to abide by to make sure that the tick doesn't escape primary containment.
Towner, sorry, Dr. Towner, there was one question in the box that I think would be a good one. He was just discussing in general the fact that we're talking about a lot of these agents that are endemic in, in regions of East Africa, South Africa, West Africa, and about engagement with locals as far as answering questions and addressing the pathogens. And I think any of us could kind of answer these, but I think from our part, I'd like to say that I think the engagement with local public health officials is a high priority and really critical, so that we can A, be asking the right questions, B, transferring knowledge, techniques, assisting with diagnostic capabilities, and really these partnerships are critical when it comes to addressing these pathogens. So that's just my short answer to that question I saw, because I think it's important.
Yeah, I can chime into that one too. I mean, we work with veterinary diseases, which may not be zoonotic, but they definitely affect human livelihoods. We've had projects, not so much in Africa, but our group specifically in Asia, so in India and Vietnam, that have FMD endemically. And these have been projects that are actually funded by the US Defense Threat Reduction Agency, so DTRA within DoD. With multiple purposes, because doing these sort of surveillance studies, for once, it gives us information on what viruses are circulating, which is good for the US and preparedness. But a big part of it has also been capacity building and collaborating with the local researchers, so that they're actually doing the work on the ground, and we're supporting them with knowledge and to some degree funding for technology. I know that just sponsored some sequencing machines and things like that. So it's a big part of what we do in our field projects.
Yeah, I mean the capacity building part is tough. There's a lot of different layers on it, from having the right tests available to the very basics of can you maintain cold chain storage. Specimen transport is an awful one. I mean, I still have yet to see that get tackled. I don't know what USDA's experiences are, but at least with CDC, it's
yeah, no, that's specifically in our field studies. That's probably the biggest issue. And what I said with Hylomatix virus, it's very sensitive and it basically falls apart when you look at it. So just getting it from the field into the lab and keep maintaining it viable is a huge problem.
I guess any other questions from any of the panelists? I'm kind of tapped out here. All right. Well, I guess maybe we'll finish just a little bit early, give some folks some time back for their day. I really want to thank the panelists for excellent discussion. I think we learned a lot from your presentation. So thank you very much for all the time and effort that you put into that. And thanks everyone for in the audience for participating. I think this is fantastic that we've got, I just saw in here in the meter over 480 people at one point. So all over the world. So this is really a fantastic opportunity to broadcast the message about some of the work that's being done for these really important pathogens. And so I think tomorrow is diagnostics, same time, 9 o'clock Eastern Daylight Time. And so I would invite everybody to come back tomorrow morning for another session. So with that, I think we'll stop there. And I guess I'll turn it over to Haley if you have any final comments or anyone else has any final comments.
Thank you so much, Dr. Towner. No, that is the conclusion of today's programming, and we will go ahead and wrap it up. I will remind all of our attendees that once you X out of your windows, you will be prompted with a quick 5-minute survey. If you wouldn't mind filling that out, it helps us improve our programming, and we really appreciate you being with us. Have a good day, everyone. See you later.